So good morning. So today we'll be discussing about the general properties of virus. This is the first topic. It is the virology. First, as all of you know, these viruses are the smallest microorganisms of all the microorganisms we know. Example like bacteria, parasites, and fungi. Come to the first slide, introduction. So these viruses are very much smaller than all the microorganisms. So they can be seen only under the electron microscopy. They cannot be visualized under the general ordinary light microscope. And the cultures cannot be cultivated or isolated in the routinely used culture media. Like what are the media what we use for the isolation of bacteria and also fungi. They can be cultivated or isolated by tissue culture methods, which are done very rarely only in specialized well-equipped laboratories. So in ordinary general laboratories, so the diagnosis of viral infection is mainly based on the antigen or antibody detection, that is serological tests, or by detection of the genetic elements RNA or DNA of the viruses, that is by molecular method. Example is by PCR, that is polymerase chain reaction. So how do these viruses differ from bacteria? First thing is the viruses do not have a cellular organization, that is they do not have cell wall, cell membrane, also the cellular components like mitochondria or cytoplasm. So where the bacteria have all these components, that is they have the cell wall, mitochondria, cytoplasm, all those things. And coming to the genetic composition, so this bacteria they contain both DNA and RNA, whereas the viruses they contain only DNA or RNA but never both. So based on the genetic composition, so the viruses are classified into DNA viruses and RNA viruses. Let's come to the multiplication. So these bacteria undergo multiplication by binary fission, that is formation of two daughter bacteria from a single parent bacterium, whereas the viruses multiply by a complex process. And one more thing is that the bacteria they have their own enzymes for synthesis of nucleic acid and also for protein synthesis, whereas this there is an absent, whereas these enzymes, that is enzymes for synthesis of nucleic acid and protein synthesis, is not seen in viruses. So the viruses have to depend upon the host cell enzymes for their multiplication. Next, that is what the self-replication is possible in case of bacteria, but it's not seen in case of virus. They are dependent upon the host cell multiplication. Next, effect of the antibacterial antibiotics. So they are effective only against bacteria, whereas they are not effective against viruses. So the common reason why it's quoted is, you can see this bacterial antibiotics they act on the cell wall of the bacteria, whereas viruses don't have cell wall. Next is morphology of viruses. So morphology of viruses, so that is discussed under two subheadings. One is the size of the virus and shape of the virus. First, coming to the size of the virus. As we have already discussed in this first slide, viruses are very much smaller microorganisms. So they vary in size from 20 nanometers to 300 nanometers, and they can be seen only under electron microscopy. Next, coming to the shape of viruses. So the viruses they have three different types of shapes. One is spherical shape, tubular shape, and one more is complex shape. The most two common things are spherical or tubular in shape. Out of this, most of the viruses they are in spherical in shape. Whatever may be the shape, both spherical or tubular shape, so the viruses they are made up of structural and functional units called as capsid. This capsid is nothing but a protein which is further made up of capsomeres. So the function of capsid is, first thing, it encloses the nucleic acid, that is DNA or RNA of the viruses, and protects the nucleic acid from inactivation by various cellular enzymes, that is host cell enzymes, and also facilitates the entry of the nucleic acid, that is the viral nucleic acid, into the host cells. Next, there are three different types of symmetry are exhibited by the capsid. One is the icosahedral symmetry, helical symmetry, and the third one is complex symmetry. The most common symmetry of all this is icosahedral symmetry. What is icosahedral symmetry? Icosahedral symmetry is a polygon which has 12 vertices and 20 facets or corners. So two types of capsomeres constitute the capsid. With pentagonal capsomeres the vertices and hexagonal capsomeres are the facets. Helical symmetry means both the nucleic acid and the capsomeres are wounded together to form helix or a spiral tube or cylindrical in appearance. Next is envelope. What is envelope? Envelope is one of the most important part of the viruses, which is an outer coating portion of the virus. So this envelope is not seen in all the viruses. So there are two different types of viruses based on the envelope. One is enveloped viruses which have envelope. The other one is non-enveloped viruses, or also known as naked viruses. Why this envelope is seen only in some viruses, why not in other viruses means, so the formation of envelope depends upon the release of viruses from the host cells. So once the virus is released in the host cell, it is by two processes, either by budding or by cell lysis. When the virus is released by a process of budding, so envelope is seen. So envelope is a lipoprotein in nature, whereas the lipid part is derived from the host cells, the protein part is from the viral coated. In case of viruses which are released by lysis to the host cells, so there is no formation of envelope. Such viruses are called as non-enveloped viruses or naked viruses. As you see, this envelope in some viruses they carry two different types of structures. One is a triangular structure called as hemagglutinin spike, which helps in the fusion of the virus to the host cells. Example, in case of influenza virus, this hemagglutinin spikes helps in the fusion of the host cell to the respiratory epithelial cells. One more structure, it is a mushroom shaped spike, which is called as neuraminidase. The function of neuraminidase is inhibition of the fusion of the virus to the host cells. This process is called as elution. So one thing you should note that all viruses which are enveloped do not have these spikes. Only certain viruses which are enveloped, they have spikes. Example like influenza virus. Let's come to the next slide is multiplication of viruses. So the multiplication of viruses is described in 6 steps. One is adsorption, penetration, uncoating, biosynthesis, maturation and release. So once the virus enters into the human body, the virus before multiplication it has to absorb or it has to fuse to the host cells. So we'll see in the next slide. So this is first thing is absorption. Absorption is nothing but an interaction of virus with a specific receptor on the host cell surface. Means all the viruses they have some specific receptors on the host cells. Example like HIV virus, it has a CD4 receptor on the host cells. Only the host cells which bear CD4 receptor, the virus goes and fuses to such kind of cells. Example in case of what's told HIV, the CD4 receptor is found only in the immunological cells like monocytes, macrophages, B cells and all those things. And also in case of coronavirus, ACE2 receptor is the receptor of the coronavirus. So this ACE2 receptor is found mainly on the respiratory epithelial cells and also in the lungs. So because of this coronavirus has a significant effect on the respiratory epithelial cells and also in the lungs. So once there is a fusion, the virus to the host cell surface, the virus has to enter. It's called as penetration. So penetration is nothing but an entry of virus particle to the host cell by a process called as viropexis. It is also generally described as endocytosis. This viropexis is a process which is seen in case of non-enveloped virus. In case of enveloped viruses, they envelop the virus, fuses to the host cell and then releases the nucleocapsid into the host cells. Next step is uncoating. So once the virus is in the host cell, so the viral nucleic has to be released. So before the release of viral nucleic acid into the host cell, that is cytoplasm or nucleus, the virus has to shed its structural component, that is capsomeres. So how this virus sheds it is by the lysis of the host cell enzymes. Example like lysosomes in the host cells, they cause breakdown of this capsid or the capsomeres. So once the capsid breaks down, there is a release of viral nucleic acid into the host cell, cytoplasm or into the nucleus. This is called as uncoating. Next is biosynthesis. Biosynthesis is nothing but the synthesis of viral nucleic acid. In general form to again So this is the formation of daughter virons. So once the daughter virons they are formed means, so they have to undergo synthesis from the parent viral nucleic acid. So this is a step in biosynthesis, that is synthesis of the daughter cell, viral nucleic acid, capsid proteins and various enzymes which are required for the synthesis of daughter virons, their assembly and release. So there are various steps in biosynthesis. One is transcription of mRNA from viral nucleic acid, translation of mRNA into early proteins, replication of viral nucleic acid and synthesis of late proteins or structural proteins. So the late proteins or structural proteins contribute in the formation of capsid or the capsomeres, whereas early proteins they contribute to the synthesis of viral nucleic acid, that is DNA or RNA. So once this nucleic acid and other components like the capsid they are formed, so they have to be arranged. So this arrangement is called as like maturation. So maturation, it is the assembly of newly synthesized viral particles, that is DNA or RNA and the capsid protein that into daughter viral. This is called as maturation. So once they undergo maturation, they have to be released from the host cell. This is called as, this takes place another step called as release. Release is nothing but release of daughter virons, that is by lysis of the host cell or by budding the host cell. So when the daughter virons are released by the lysis of the host cell, there is the formation of non-enveloped viruses or naked viruses. And then the viruses are released by budding of the host cell, it results in the formation of capsulated virus. So this is the diagrammatic representation, this viral replication. The first picture you can see the fusion of the virus to the host cell. Next is penetration. So you can see the green dot. So it is the virus which is penetrated in the host cell cytoplasm and then to the nucleus. So next it undergoes uncoating, that is removal of capsid. So by the lysosomal enzymes in the host cells. So once these are released, so the viral nucleic acid enters the nucleus and these undergo synthesis, that is to form the daughter virons. So in the fourth picture you can see the formation of large number of nucleic acid and also the capsid proteins. So in the next picture there is a fifth diagram you can see. So there is assembly of this daughter virons and viral nucleic acid to form daughter virons, or also called as progeny virons. And next picture, that is the sixth picture, you can see the release of the daughter virons or progeny virons in the host cell. Thank you.